Hi folks. Today we present a vital and visually fascinating biological process from a number of points of view. Those of biology, chemistry, physics, and math. I'm Professor Charles Walker, a faculty member of Molecular, Cellular, and Biomedical Sciences at UNH, and I'll begin by describing the process from a biologist's perspective. The molecular motor kinesin is an ATP-driven biological machine that carries vesicles and individual proteins from near the nucleus to the cell surface on microtubular highways formed from the polymerized protein tubulin. This delivers proteins and vesicles from near the nucleus to remote sites within the cell, a distance of 8 to 20 micrometers that doesn't seem like much but cannot be accomplished by diffusion alone. In this animation that I'm about to show, there are three things to observe. The animation begins with monomers of the protein tubulin assembling or polymerizing into a microtubule. Next, the animation shows similar monomers disconnecting and dispersing in the cytoplasm as a microtubule is disassembled. Microtubules are part of an ever-changing array that extends throughout the cytoplasm between the nucleus and the cell membrane in each of your cells. Finally, observe the molecular motor kinesin walking on a single microtubule with two foot-like extensions on one end. These are ATPases that hydrolyze ATP and provide the energy for the steps that tubulin takes. And they have a fluid-filled vesicle attached to the other end. Vesicles carried by kinesin can contain different types of sugar-coated proteins destined for various cellular locations. For example, sugar proteins synthesized in the rough endoplasmic reticulum are sorted by the Golgi apparatus and emerge from the exit surface of the Golgi, the so-called trans site, in vesicles targeted to distant places within the cell. A vesicle might contain sugar-coated proteins to be expelled from the cell, such as hormones or components of the extracellular matrix that surrounds and protects the cell. Alternatively, vesicles might carry signal recognition molecules that serve as receptors to be inserted into the membrane of the cell, rendering it sensitive to the outside world. In the rest of this presentation, we'd like to show you how related chemistry, physics, and math can help explain the action of kinesin and its importance in the cell as an alternative to diffusion.
Hello, I'm Chris Bauer, and I teach general chemistry at UNH. Here are aspects of the chemistry responsible for kinesin movement and function. This animation of the walking action of kinesin shows the active end of a single kinesin molecule, emphasizing the feet that interact with a microtubule. Each foot is actually an enzyme called an ATPase. This enzyme hydrolyzes the molecule adenosine triphosphate, or ATP, releasing a single phosphate group and leaving behind ADP, adenosine diphosphate. In this process, energy is released and the shape of the foot changes, allowing it to take a step along the microtubule. The foot in this altered shape is actually a ligand that will bind to receptor subunits of the tubular protein, securing it briefly to the microtubule. Here is a molecule of ATP. These phosphates each carry a negative charge. Their close proximity tends to make ATP unstable and ready to release one or two of the phosphates. This happens by a reaction with a water molecule called a hydrolysis reaction. The ATPase enzyme surrounding the ATP in the kinesin, foot, promotes this reaction. The structures of the reaction products are more stable than the structures of the starting materials. The stepping motion is a fascinating sequence of attachments and detachments of molecular segments to and from each other. This results in conformational changes, slight rotations in covalent bonds in the backbone of the protein, or movement in the relative positions of the segments of the protein. Most of these attachments and detachments involve intermolecular forces, such as ion-ion attraction, ion-dipole attraction, hydrogen bonds, and nonpolar dispersion interactions. The type of interaction depends on the amino acids in those locations, in particular on what are called the side chains of those amino acids. Some carry a charge, like glutamate with a negative carboxylate group, and lysine with a positive ammonium group. Others can hydrogen bond, like serine because of its OH side chain. The binding or loosening or conformation change happens because the protein framework is moving to the most stable three-dimensional arrangement to maximize attractive forces. Subtle adjustment of one part of the structure to make a stronger attachment leads to a detachment in another part of the structure because the conformational movement causes the intermolecular forces there to weaken, which in turn causes further mechanical motion of the protein, and so on. ATP plays a special role. When the loose foot snaps into place by intermolecular attractions between the foot and the microtubule, this causes strain in the second foot. This strain energy increases the likelihood that the weak OP bond in the ATP in the second foot will be broken by reacting with adjacent water molecules. Breaking of that covalent bond allows substantial conformation change such that foot 2 is released from the microtubule. It is ready to move forward for the next step. Notice that one ATP molecule is involved in each step and it must bind to the foot to trigger the next step. The video suggests that the ATP knows where to go, but it doesn't. ATP is present in the cellular fluid and through random motion gets close enough to the binding site to be drawn in by the intermolecular forces.
Hi, my name is Dawn Meredith and I teach the Introductory Physics for Life Science students here at UNH. And I'd like to talk to you about kinesin from the physics point of view. What do physicists like to look at? We like to look at position, velocity, acceleration, forces, and energy. And today we will focus only on position and velocity. Let's think about why molecular motors are needed when diffusion might do just as good a job at moving vesicles around the cell and do it with far fewer resources. Diffusion is the mechanism for moving molecules around inside a cell nucleus. So why doesn't diffusion work so well to get a vesicle from the center of the cell to the edge? Let's look at this with some numbers. Taking some realistic values, let's say that the size of the nucleus is equal to 3 micrometers. And let's say that the size of the cell is equal to 12 micrometers, which is 4 times the size of the nucleus. Because diffusion is a random process, we know that the time it takes to travel a certain distance grows as the distance squared. So it's equal to the distance squared divided by the diffusion constant. And we'll take the value of 30 micrometers squared per second, which is typical for cells. And this squared value right here turns out to be very important. We can use this relationship between the distance traveled and the time to compare the time to diffuse across the nucleus to the time to diffuse across an entire cell. So the time to diffuse across a nucleus is equal to that distance across the nucleus, the 3 micrometers, and that whole thing squared divided by the diffusion constant. And remember, we have to square everything inside of there, so that's 9 micrometers squared divided by the diffusion constant, which gives us 0.3 seconds. And we can do the same thing for the time it takes to diffuse across the cell. We take the 12 micrometers for the cell size and divide it by the same diffusion constant. And we put those numbers in our calculator and we get 4.8 seconds. So it takes considerably longer to traverse the cell, which is expected. In this situation, we have a different relationship between the distance traveled and the time. Here we have the distance traveled is the velocity times the time. And here the distance traveled and the time are linearly related, so that if the distance is 4 times bigger, the time is just 4 times bigger, not 16 times bigger as it was for diffusion. But you can see this more clearly if you make a plot. And what we'll plot here is the time to travel versus the distance traveled. And here I'll make a plot for diffusion, which goes like the square root of the time, versus constant velocity, which grows linearly. And as time goes on, you can see that the difference between the diffusion and the constant velocity increases. So here it's small, and here it's bigger. So this little thought experiment helps me understand why molecular motors are needed for transportation, even for very small distances.
So what does all this mean? It turns out that these molecular motors are of great interest these days to nanotechnologists, because what we have here are actually little nanomachines that people understand from a chemical, biological, and mathematical point of view. They know how much these things can carry, like kinesin. They know how far they can walk. They actually know that some of them limp. And interestingly, for nanotechnologists, these things are based on DNA, so they're programmable, and you can actually change their function. Some people have stretched out microtubules in a dish, put in biologically modified kinesin, had it carry something from one end of the dish to the other and drop it off. All you had to do is add ATP. So they're pretty interesting molecules from a number of points of view.